I'm Sandy Macrae. I'm the CEO of Sangamo, Sangamo Therapeutics, and I'll refer you to our forward-looking statements. And Sangamo is a new company that's come out of a much older story, a much longer story, and reflects the need to do all the development processes to take a scientific idea and make it into medicines. And this year we will have 9 clinical trials in play, and we are becoming a genomic medicines company. But we're committed to translating groundbreaking science into these genomic medicines. And so what I want to tell you about now is a range of therapies, a range of technologies that take you everywhere from gene therapy to cellular manipulation and cell therapy, and to the future, which will be genomic editing and finally gene editing. Because as one of the advantages that we've gained with that time with Sangamo is that we can do a range of technologies. And I take it from the left-hand side. Gene therapy is now. Gene therapy is where many other companies are at this moment. But with our Hemophilia A partnership with our friends at Pfizer, a great relationship that we have there, we are in the world of now. Hemophilia A and the treatment of that as a gene therapy is now something that we're all accepting as the norm. And if you look at many of the patients in this area, they've stopped taking factor. Their condition has been resolved, and people whose lives were dominated by daily injections or several injections a week now have a solution that will last them many years. And then the second, and that world, if we can be successful with Hemophilia A, it reads forward to our Fabry program and a whole list of other gene therapies that we have in the pipeline. And then next to that is ex vivo genome editing. And this started with the work Sangamo did 12 years ago on HIV. And I'm sure a number of you saw the second Berlin patient. It's actually London, but London's no longer in Europe, so we've got to call it the Berlin patient. And that implies that CCR5 editing will be able to resolve, has a potential to resolve HIV. And Sangamo did work in that, and then we built on it with beta thalassemia and sickle, partnered with our friends now at Sanofi. Again, a good relationship, and I'm going to come back to that because it's really important to have successful partnerships when you can't do everything yourself. And that will hopefully allow those conditions to be resolved by editing one single part of the genome, will provide the fetal hemoglobin that will prevent these patients having transfusions. And then we're going to expand that. We bought a company in France called TXL, which allows us to edit Tregs. And what it speaks to is the range of things that we can edit. With Gilead and with oncology, we're editing CAR-Ts, and we're providing them state-of-the-art editing capability to choose oncology solutions that are autologous now, will become allogeneic. With the CAR-Tregs, we're starting off in a transplant indication, but we're going to be moving rapidly to autoimmune. And we hope that CAR-Tregs is going to be the next kite and the next Juno, and will provide a solution for autoimmune disease. And then finally, on the far right of this slide is in vivo genome editing. And then finally, it will be gene editing, transcription control. That is by far the most difficult thing, and it's far the most difficult thing that anyone's trying in the whole field of gene editing. It's easier to do it when you take the cells out of the body, you edit it, you know exactly what you're giving back, and you put it back into the patient. With gene editing and genome editing, you have to rely greatly on delivery. But the target, the quest is a good one, because eventually if we can give each one of you a single injection that would help you address the disease that you suffer from, that is the final goal of gene editing, and it's final goal of a genomic medicine company. And so we have a pipeline, as I said, that will have 9 things in the clinic this year. We, on the top in our inherited metabolic diseases, we have MPS I and MPS II, and we showed data at World Symposium earlier this year, where we feel we've got the finger holds on the start of that journey to genome editing. We showed editing of in the liver of these patients. We showed some expression of enzyme. We showed some uptake in tissue. And with the second generation of zinc fingers, we hope to be able to take that to the next stage. Fabry disease, the IND's approved. It will be going into the clinic with this later this year. In hematology, we have our Heme partnership with Pfizer and our thalassemia and sickle cell partnership with Sanofi and ex-Bioverativ. We also have a preclinical program, and these are things that will come into the clinic during the year. With Kite Gilead, we have a CD19 that they hope to have as an IND this year, and we have a deal with them for 10 different targets that will allow us to be their editing machinery and it will allow them to fulfill the dream they had when they purchased Kite. And then for immunology, we have the Treg TXL company, and that's something that I think is beneath the radar, below the radar for many of you, and it really is a fascinating future. And finally, for CNS, we have a single compound deal with Pfizer for C9orf, which offers the promise of being able to control one of the causes of ALS, and a Huntington disease deal with Shire, now Shire Takeda, that hopefully will be able to resolve what is an awful disease. And finally, on this slide is the tauopathies, and with the announcement today of Biogen and the end of the amyloid hope, we believe that this gives us a real opportunity to turn down that protein. So I'm going to finish with this slide before I sit with Amanda and why we believe that there's great hope for Sangamo. We've spent the past 2.5 years since I joined building a management team, building an infrastructure, all the things that make a company reliable and able to produce and develop the science. We have the leading genome technology. There was a paper that went out this month, I believe, that will describe the new way that we can modularize the zinc fingers, and we'll have a paper coming out later in the year that shows how they're the most specific. We have the clinical expertise to move things forward into the clinic. We have a strong balance sheet, and importantly, we have good partners where that's appropriate. We will take medicines forward ourselves and be a fully integrated company. But where we can't do that and there is scientific promise, we will do that through partnerships with the Pfizers, the Gileads, the Sanofis, and any of the rest of you that are in the audience. But it will only be for strategic partnerships where we can really get the medicines to the patients quicker. So I'll pause there and happy to take your questions.
Thanks, Sandy. I actually thought we maybe would start with the cell therapy side or the ex vivo part of the business. So you talked about the relationship with Gilead and in the allo CAR T space. One thing it seems quite obvious in an allo perspective that you're going to have to do sort of multiplex editing, something that some other editing platforms have talked about, their ability to do that. I was wondering if you could just walk us through kind of what data you've shown on that front, or what you're able to do in terms of multiple knockouts.
Happy to do so, because it's important to truly use the full capability of editing. So at the time of the deal with Gilead, one of the things that they asked us if we could do would be to allogeneize a cell, put in a target, and then do something else. And so we showed data where we took out beta-2 microglobulin, we took out the T cell receptor, dropped a new CAR into that locus, because I think putting in the locus allows a degree of control, and we edited CISH, so it's another edit, so it was a quadruple edit, and it was at the level of about 70%. And how you get to that number is compound interest, we call it. So you've got to be sure that each one of the things you do happens at a sufficiently high frequency, so as they all happen at a frequency that is useful. So we're now, when we edit T cells, it's about 90, 95, even in one case 99% editing efficiency, making sure that we reduce off target and being very careful that there's no cross interaction between the edits.
I think one of the other questions is just in terms of the more engineering you're doing, does that impact the fitness of the cell? Curious what you're seeing, especially as you do multiple knockouts.
It's a great question, and to do those edits, we use Macrae as how we electroporate the cells. So we do it once, and that keeps the cell fit. And so you've really got to be able to do it all in one shot. We've done 4. We've played with more than that. So far, we haven't needed to do more than that. And the fitness of the cell is involved in how you process them and deal with them later, which is why we chose to buy TXL, because we could do the editing, but what they were really good at was looking after Tregs, and they had put together a process for how to do it. So there was a really nice coming together of the two companies. We needed their cellular knowledge, they needed our editing.
Which is a perfect segue into my next question on TXL in terms of, obviously many people are using many different cell types. You mentioned Tregs kind of under the radar. What was the background behind why you chose to go with Tregs versus some other type of cell?
So the deal with Kite Gilead, is that they get T cells and NK cells for oncology, and that allowed us to look at seeing it's so much easier to do ex vivo editing. What are the other cell types? And there's a whole range of things, everything from B cells, macrophages, iPSCs. When we looked at T cells, which is the thing we know best, we noted the literature on polyclonal Tregs, and there was a great hope for that. And we felt that that was the next near thing. We felt that the experiment hadn't been properly done yet because no one had done a directed therapy. And the reason I like it is because you don't need to know the causative antigen, you need to know a localizing antigen. So if you're going into the CNS, you need to know an antigen that gets you to myelin, for example, for MS. You don't need to know the cause of the MS. And once it's in the area, the Treg gets activated, and no matter what the causative antigen is, it calms down the local immunological reaction. So it allows a greater variety of diseases to be targeted. I mean, you could be going for types of collagen in a joint for rheumatoid arthritis, things that will take you to the gut for Crohn's disease. So we're very excited to move this ahead. We've now fully integrated the two companies. We've brought their CEO in-house as our head of corporate development to make sure there's a tie between us both. And we look forward to a CTA for TX200, which is for transplant mismatch, which will act as a test bed and hopefully also provide benefit for those patients and perhaps even a commercial product there.
And you haven't given any timing around that?
CTA this year, we said.
This year, okay. Also wanted to ask, so there's been a lot of discussion around MPS, and as a part of that, you talked about sort of the next generation of zinc fingers. Mm-hmm. Obviously, you've been working with them for many, many years, and you've published some papers around what you're doing with the nuclease, et cetera. Just wondering if you could talk a little bit about that to the group, and then how are you rolling that into your portfolio?
Gene therapy is now, and we have a gene therapy portfolio. Ex vivo is coming, and we are investing greatly in ex vivo. In vivo editing is the most difficult thing we do. We are constantly upgrading our technology, and so for the second generation zinc fingers, we alter the 5', which alters the translation efficiency, the 3', which alters the stability of the message, and we increase the avidity, and with that we get 5 to 30-fold increased cutting. They also wobble over a SNP, so we can go into more patients, and the off-target, we've reduced some of the off-target. So it is a product line extension. It's our responsibility to constantly make our technology better. We recognize that the MPS data wasn't good enough for those patients. We need to see the data over the next coming months and then work out where we apply the second generation. We hope to have data from that in 2020 to allow us to make a decision about going into phase 3.
We have 15 seconds. I thought maybe you'd talk a little bit more about Fabry disease, given that you just got the IND. Have you talked about kind of the trial structure yet, or when exactly that might?
So I have 5 seconds.
Go.
And the IND's approved. The clinical sites are in discussion. We will be in the clinic with us at the end of the year.
Perfect. All right. Thank you very much.
Thank you.